Hello and welcome to Nikolai's genetics lessons, and today's video going to be about forensic genetics. And here's a problem. A group of bodies are found buried in a forest. The police suspect that they may include the missing Jones family, 2 parents and 2 children. They extract DNA from the bones and examine using PCR genes A and B, which are known to contain tandem triplet repeats of variable length. They also analyze DNA from 2 other men. The results are shown below, where the numbers indicate the number of copies of tandem repeat in a particular allele. For example, male 1 has one allele with 8 and another allele with 9 copies of tandem repeat in gene A. Could the woman have been the mother of both children? Why or why not? Which man, if any, could have been the father of the child 1? Let me explain a theory first. So imagine that this is chromosome number 1 and this is also chromosome number 1. We have all our chromosomes in pairs because we got half of our chromosomes from our mother and half from our father. And say somewhere here we have a locus. Usually such loci are outside the genes, where we have different number of tandem repeats. Because this loci outside the genes, usually mutations happen here and we have different number of tandem repeats here. What tandem repeat means, for example, A, T, T. Such sequence can repeat in this locus. In some people, say 2 times, and in some people 3 times. For example, A, T, T, A, T, T, A, T, T. So as you see, we may have in the same locus, whether say in our example 2 repeats or 3 repeats. Let's say we inherit 2 repeats in this locus from our mother side and 3 repeats on the homologous chromosome, which we inherited from our father side. Here we also have 5' end flanking region and 3' end flanking region. And if we prepare primers, which are going to be the same, because this region is stable and doesn't have any mutation, so we use the same primers, but the product would be different, because in one case we have 9 bases here, and here the product of PCR would be multiplication of these 6 bases. And if we with second stage would run gel electrophoresis, what we are going to get? If we load this product here and run a gel, we are going to get 2 bands, because those products that is going to be smaller products of PCR would run through the gel faster and would travel longer distance. And those which are bigger, these molecules, would move slower. So we would have 2 bands. Also, when we run this gel, we preload here a ladder. This is molecules of the known size. For example, we know that here we have, say, 4 repeats of this triplet nucleotide, and here 3, 2, and 1. So just comparing these bands, we can say that here we also have 3 repeats, and here we have 2 repeats. So this person has 2 and 3 these numbers that specify this locus here, 3 and 2. It also can be 2 and 3, because we don't have left and right. We just know that here in this locus on the chromosome number 1, this person has 2 repeats and 3 repeats of trinucleotide. But what if person would have only one band? Can this happen? Yes, this may happen. For example, at the different locus, this person may have only one band. What does it tell us? This tells us that at the different locus, this person has the same allele inherited from both of the parents, meaning mother at this locus has, say, 3 repeats, and father at this locus also has 3 repeats of this trinucleotide, and we would see only one band. We also say that this person is homologous at this locus. And when we do genetic fingerprinting of the person, we compare not only one locus, but we compare about 13 up to 20 loci, and at each locus we may have up to 20 alleles of the different sizes. So when we say making genetic fingerprinting of the person, we can say that at locus 1 we may have 20 variants. At locus 2 we may have 15 variants. At locus 3 we may have say 25 different combinations, and so on. And now we have to multiply all these probabilities of different combinations, and if we will do it for each locus, and as I said, we usually analyze between 13 and 20 loci, we can get such astronomical number that exceeds number of the people on our planet. Now let's return to our problem. Could the woman have been the mother of both children? So here is a woman, here is a children. We know that each child would inherit one chromosome in each pair from mother side, another from the father side. So at least one number should come from mother side. Mother for the gene A has 8 repeats on both of her chromosomes. So her child have to inherit 8 repeats. And we see that for the gene A, child number 1 has 8 repeats, which he can inherit from the mother side. And child 2 also has 8 repeats, which he can inherit from the mother side. Let's now analyze as for the gene B. And we see that woman has 3 and 5 repeats at this locus. And actually this is not a gene. We would better say this is locus B. So gene meaning that the sequence would make a protein or specify protein, code for protein. But usually when we analyze this loci doesn't specify any protein. So woman has 3 and 5 repeats, so child 1 has 7 repeats and 5. So this woman can give this 5 repeats for the gene B to the first child. As for the child number 2, child number 2 has 3 and 7 repeats. Again, mother for the gene B can give this child 3 repeats. So mother or this woman can be mother of both these children. We cannot exclude her. Now let's return to the second question. Which man, if any, could have been the father of child 1? So now let me underline this child. So let's compare, for example, male number 1. Could male number 1 be a father of the child 1? For example, we know that woman can give allele with 8 repeats, so the child have to inherit 7 repeats from the father. And male 1 for the gene A cannot give this 7 repeats. But you may say he can give 8 repeats. But that means that 7 repeats this child should get from the mother, and mother have only 8 repeats. So we can exclude male number 1 as a father of the first child. So now let's analyze male number 2. Again, woman can give only 8 repeats to this child. So this 8 reserved for the mother. And we expect that child should get 7 repeats from the father. And again, male number 2 only can give 8 or 6 repeats. So we also can exclude this male as possible father of this child. Now let's analyze male number 3. Mother only can give 8 repeats, and 7 repeats child number 1 have to get from the father side. And we see that on one of the chromosomes, male number 3 has 7 repeats. So here can be for the gene A or locus A, provide 7 repeats to his child. So child can inherit this chromosome from him. Now let's analyze gene B or locus B. And we see that child has 5 and 7 repeats, woman has 3 and 5. So these 5 repeats this child can inherit from his mother or from this woman. And from his father he have to inherit 7 repeats. And we see that male number 3 has is homozygous for this locus and has on the homologous chromosomes the same number of repeats. So if we would run gel electrophoresis, we would see only one band here, and here can also give this any of this chromosome child would inherit and would get 7 repeats. So we can say that male number 3 can be father of the child number 1. And this is exactly how paternity test is working. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. And see you in the next video. Goodbye.